Dark phase reactions in different plants take place in three different ways: C3 cycle or Calvin cycle, C4 cycle, and Crassulasian acid cycle or Crassulasian acid metabolism. Now, let us try to understand the C4 pathway. Until 1961, plant physiologists thought that there is only one way of carbon fixation, that is Calvin cycle, C3 cycle. There is no other way. All the plants fix the carbon dioxide through the Calvin cycle, C3 cycle. But in 1961, Kortschak and Burr, they have given the hint that there may be other type of carbon dioxide fixations. They reported that in some plants, the first stable compound in carbon dioxide fixation is not phosphoglyceric acid, but C4 compounds like oxaloacetate or malic acid or aspartic acid. Later, two Australian plant physiologists, Hatch and Slack, worked out the details of the new pathway. They have completely worked. These people gave the indication, but the details are worked out by Hatch and Slack. Hence, on their name, the C4 pathway is also known as Hatch-Slack pathway. It is also known as C4 dicarboxylic acid pathway. The first stable compounds are C4 compounds. They contain two carboxyl groups, dicarboxyls. Hence, C4 dicar. It is an alternative pathway to C3 pathway. C4 cycle has been reported in 19 genera. Majority of the plants C3 cycle, but C4 cycle, 19 genera, 90 species belong to 11 families. Those families, some belong to monocots, some belong to dicots. In monocots, Gramineae, Poaceae, just like sugarcane, Jowar, maize, pearl millet, then finger millet, Cyperaceae members. In dicots, three families, Amaranthaceae, Portulaceae, Boraginaceae, these families contain the C4 plants which fix the carbon dioxide through alternate pathway. In this pathway, important, CO2 acceptor is phosphoenol pyruvate, PEP, commonly known as PEP. Carboxylation is catalyzed by PEP carboxylase. The first stable compound is oxaloacetic acid, OAA, C4 compound. In this pathway, both C3 and C4 pathway operate. Now, when we compare the most important differences between the C3 pathway, C4 pathway, in C3 acceptor is RUBP, but in C4 it is PEP, phosphoenol. There the enzyme is Rubisco, here PEP carboxylase. The first stable compound there, PGA, phosphoglyceric acid, but here C4 compound, mostly oxaloacetic acid. In C3, only C3, but here in C4 plants, both C3 and C4 pathways operate, one dependent on another. Then in C4 plants, if you take the leaf, take the section, thin sections, observe under the microscope, then you will find a peculiar anatomy called cranch anatomy. Only in C4 plants the leaves show the cranch anatomy. What is that cranch anatomy? Around the vascular bundles, these vascular bundles are found in the vein regions. Around the vascular bundles, some specialized cells are there. These are called bundle sheath cells. Here, these bundle sheath cells around the vascular bundles are absent in pure C3 plants. Only C4 plants show the cranch anatomy. Speciality of the cranch anatomy, bundle sheath cells around the vascular bundles. They also contain the chloroplast, just like the mesophyll cells. Epidermis, upper epidermis, lower epidermis, then stomata, all these will be there, but only peculiarity is the presence of bundle sheath cells around the vascular bundles. Now, cranch anatomy. All C4 plants exhibit specialized anatomy called cranch anatomy. These plants possess bundle sheath made up of large sized cells around the vascular bundles. The chloroplasts of mesophyll and bundle sheath cells are morphologically different. Chloroplasts are present both in mesophyll cells and here in bundle sheath cells, but morphologically they are different. So therefore, chloroplasts in C4 plants are dimorphic, two types, one here, another here. Mesophyll cells chloroplasts are normal with grana, but bundle sheath chloroplasts lack the grana. This is another important point to remember. They contain starch granules. Thus, chloroplasts in C4 plants are dimorphic. The primary function of the cranch anatomy is to provide a site, a place in which CO2 can be concentrated around the rubisco, thereby avoiding the photorespiration. So rubisco is there. This rubisco can it accept oxygen, it can accept carbon dioxide also. Whichever is more in concentration, that acts. So in avoid to the enzyme to be in contact with oxygen, this mechanism operates in order to avoid the photorespiration. In the next lecture, we will see what is photorespiration. Mesophyll and bundle sheath cells are connected through numerous cytoplasmic sleeves called plasmodesmata. I will show you the diagram. There are 4 phases in C4 cycle. 1. Formation of C4 acids, especially oxaloacetic acid, with the help of PEP carboxylation. Carbon dioxide acceptor is PEP, enzyme is PEP carboxylation. Transport of C4 acids, the C4 acids are formed here. These C4 acids are transported to bundle sheath. Decarboxylation means coming out of the carbon dioxide. Suppose one compound gives rise to carbon dioxide, that is called decarboxylation. Of C4 acids to release carbon dioxide, which is fixed as carbohydrate in C3 cycle, just like C3 cycle. C3 acid pyruvate formed in decarboxylation moves back to mesophyll, helps in PEP regeneration. So this is what? Here, this is normal mesophyll cell. This is bundle sheath cells connected by plasmodesmata. These are the plasmodesmata. Mesophyll cell, this is bundle sheath cell. Atmospheric carbon dioxide, when dissolves in water, it forms the carbonic acid. It is fixed by PEP, forms C4 acid OAA. Then this OAA, oxaloacetic acid, is transported to bundle sheath cells. There, the decarboxylation, the C4 acid splits into carbon dioxide and C3 acid. One carbon here and 3 carbons here, C4. C4 acid splits into one carbon dioxide and one C3 acid that contains 3 carbons. So this carbon dioxide will be utilized for the fixation, for the synthesis of glucose, just like C3 pathway. Just like C3 pathway. Fixation by Calvin cycle. So Calvin cycle reactions take place in bundle sheath cells in C4 plants, remember, in C4 plants. Then the C3 acid is transported back to the mesophyll cells and utilized for regeneration of PEP, phosphoenol pyruvate. Atmospheric mesophyll cell, bundle sheath cell, connected by plasmodesmata. Atmospheric carbon dioxide is fixed by PEP, C4 acid OAA, oxaloacetic acid. That acid is transported to mesophyll cells where decarboxylation, carbon dioxide comes out from the acid. It is utilized for the carbon fixation and synthesis of carbohydrates, just like C3 plants. This C3 acid is transported back to bundle sheath cells, to mesophyll cells for regeneration of pyruvate. Types of C4 plants. In C4 plants, further types are identified. One is NADP ME type, malate formers. Here the first stable compound is malate. Then in some NAD ME type, first stable compound is aspartate, amaranthus. Here it is sugarcane maize. PEPCK type, PEP carboxylase type, example panicum and chloris. Then Crassulasian Acid Metabolism, CAM pathway. This is a type of C4 cycle, but in few plants. Is Crassulasian acid metabolism is an adaptation to increased efficiency in the use of water, and so is typically found in plants growing in dry conditions, arid conditions, xerophytic conditions. Mostly they are xerophytes, succulents. CAM plants are succulents, but all succulents are not CAM plants. These plants, as an adaptation to xerophytic life, dry conditions life, close their stomata during daytime to minimize, to avoid the transpiration, thus by loss of water. Close the stomata during daytime, open stomata during night. These plants, CO2 fixation takes place in night time. The CO2 fixation takes place during night time when the stomata are open. Hence, this is called dark carbon assimilation. But synthesis of carbohydrates takes place during daytime. So what are the Crassulasian plants? 20 angiospermic family: Crassulacea, Cactaceae, Agaveaceae, Orchidaceae, Portulaceae, Euphorbiaceae. During night, A plant employing calf has its stomata open because exchange of gases is required, carbon dioxide and oxygen. So they open the stomata during night. So they are happy during night because there will be no light, no transpiration. Allowing CO2 to enter and be fixed as organic acid, a PEP reaction similar to C4 pathway. During night, it is similar to. Resulting organic acids are stored in vacuoles for later use. Then C4 organic acids are formed during the night, stored in the vacuoles, and they are utilized during the daytime, as the Calvin cycle cannot operate without ATP and ADP. So why the carbon dioxide is not fixed during the night means during the night there is no light, there will not be availability of the ATP and NADPH. So therefore, until the ATP and NADPH are available during the daytime, they wait for carbon dioxide fixation. During day, what happens? The stomata close and conserve water, and the carbon dioxide storing organic acids are released from the vacuoles of the mesophyll cells by an enzyme and release the carbon dioxide which enters into the Calvin cycle, so that photosynthesis takes place during the daytime. So here during daytime, when the light is available, during the night they absorb the carbon dioxide, but they do not fix the carbon dioxide. During daytime they fix. Then CAP pathway summary. Crassulasian acid pathway summary. What the salient features? CAM plants live in very dry condition. They are xerophytes under very dry condition. So they have the problem, water problem, excess transpiration. To avoid the transpiration, they close the stomata during the daytime, open them during night. Unlike other plants, open the stomata and fix only at night when stomata are open. Like C4 plants, they use PEP carboxylase to form oxaloacetate. Oxaloacetate is converted to malate within which is stored in cell vacuoles. Oxaloacetate is converted to malate. During day, when the stomata are closed, CO2 is removed from the stored malate and enters the Calvin cycle. Decarboxylation takes place. So this is how we can see C3 plants, C4 plants, CAM plants. In C3 plants, direct carbon dioxide is fixed through Calvin cycle. Direct, straightforward. But here in C4 plants, carbon dioxide in mesophyll cells, carbon dioxide is fixed. C4 acid, carbon dioxide, then it comes to the bundle sheath cells where Calvin cycle reactions take place. Then in CAM, CO2, carbon dioxide, atmosphere, C4 during night, during daytime. During night, this C4 is oxaloacetate, later converted to malate. Then it enters into the mesophyll cells. There the Calvin cycle reactions take place during the daytime. Calvin cycle during daytime. Here Calvin cycle reactions take place both during daytime and night, but here only during daytime. These reactions, so these are just like the nocturnal animals. So because they grow in dry conditions, xeric conditions, transpiration is the problem. To avoid the loss of water, the stomatal mechanism. So it is an adaptation, so that carbon dioxide is taken during night when stomata are open. But during night, what happens? No ATP, no NADPH. So they wait until the daytime when the stomata are closed, and carbon dioxide is utilized for fixation through Calvin cycle. So there are some C4 CAM intermediate species, both Peperomia, Camptotricha, Poaceae oleracea, Poaceae grandiflora. The two pathways of photosynthesis can occur in the same leaves but not in the same cells. Same leaves but not in the same cells. And the two pathways cannot couple and can only occur side by side independently in the same leaf, the same mesophyll, but cells are different. They are independent, not dependent on another. Then CO2 compensation point. So during photosynthesis, carbon dioxide is taken in, oxygen is released. Whereas in respiration, oxygen is taken in, carbon dioxide is released. In a plant, usually the photosynthesis and respiration take place simultaneously, side by side. So in majority of the cases, whatever the carbon dioxide that is released in the respiration will be utilized for photosynthesis process, and similarly whatever the carbon oxygen released in the photosynthesis will be utilized for respiration. Then how much oxygen released, how much carbon dioxide taken? The concentration of carbon dioxide at which the rate of respiration release of carbon dioxide. In respiration, carbon dioxide is released. And the rate of photosynthesis, carbon dioxide utilization, are equal, called carbon dioxide compensation point. That means rate of release of oxygen during photosynthesis, utilization of carbon dioxide in photosynthesis are equal. This is called compensation point. Again, concentration of carbon dioxide at which the rate of respiration, release of carbon dioxide, and rate of photosynthesis, carbon dioxide utilization, are equal, called carbon dioxide compensation point. At carbon dioxide compensation point, both are equal. The net gain of photosynthesis is 0. So this has a lot of impact on the productivity of C3 and C4 plants. In CO2 compensation point is less in C4 plants, more in C3 plants. So as a result, what happens? The productivity of C4 plants is more than C3 plants. So these are the differences between the C3, C4 CAM. Leaf anatomy, no cranch anatomy, no cranch anatomy. This you remember. In C4 plants, CAM plants, no cranch anatomy. Enzyme RUBPcase or Rubisco, PEPcase. Acceptor RUBP, PEP. First stable compound PGAC3, here OAA, malic acid, aspartic acid, here mostly malic acid. Photorespiration present absent. This is again one reason why the productivity of C4 plants is more, because it is in C4 plants, CAM plants, the photorespiration absent. The carbon dioxide carbon fixation. So these are again carbon assimilation rate less, more, less. On the whole, in among the three C3 plants, C4 plants, CAM plants, the carbon assimilation rate is more. Temperature, usually these grow in tropical areas. Inhibition of photosynthesis leaves transpiration ratio more, less, very less. Growth rate in plants, this is important, less, more, very less. Succulent plants growth rate is very less. In C4 plants, productivity growth is more. In next lecture, we will see about photorespiration, the respiration that takes place when there is light induced respiration called photorespiration. So what is the mechanism, what is its impact, in which plants it is present, C3 or C4 or CAM? This we will understand in the next class.